This is Brain Ponderings, conversations with neuroscientists at the forefront of brain research. And the topic of today's podcast is human neuroassembloids building brains in the laboratory, or at least parts of brains. And my guest is Sergiu Pasca. He's professor of psychiatry and behavioral sciences at Stanford University, where he's also member of the Wu Tsai Neurosciences Institute, director of the Physician Scientist Advanced Mentorship Program, and director of the Stanford Neuroscience Stem Cell Core. His lab has made major advances in developing technologies which are getting pretty sophisticated to try to reconstruct human neural circuits uh, in 3D, essentially what he's doing. And then also seeing, can you connect different brain regions in these 3D cultures and then study them, interrogate them at the molecular level, the electrophysiological level, whatever, using optogenetics, calcium imaging, which is something I was one of the, I was one of the first ones. My lab was one of the first ones to do any calcium imaging in neurons back in the 1980s, right after Roger Chen uh, made FIRA 2, because Roger knew the head of my mentor when I was a postdoc. And so, but anyway, that's the side. So welcome, Sergio.
Thank you so much for having me. It's really great to get together and have this conversation.
You're born in Romania, is that right?
I am. I was born and raised in Romania. Actually, did most of my training there, my clinical training, and then came to the United States to do a postdoc. Essentially, exactly 15 years ago, I was like packing up to come to the United States around this time, and started in January of 2009 my postdoc at Stanford.
We've accomplished a lot since then.
Been a really exciting time. I mean, I must say, I guess, there's times in science where, just the new technology just makes so many things possible so quickly. And so I always like look as being so lucky to be in the early days of like using stem cells to model disease. And again, and we can talk about that, but that's mostly anchored essentially in my clinical ambitions for psychiatric disorders.
And you, even back when you were at Romania, and then you were at Max Planck, learning some electrophysiology. You had an interest back then in human developmental brain disorders like autism and some other ones. And
Schizophrenia
actually,
as
I look
well.
back. Of course, we go on PubMed. I look back and we had some overlap in our interest a little bit, because you were studying homocysteine and one
Yes.
carbon metabolism and folate, and
Yeah.
folic acid deficiency leads to abnormalities in the development in the central nervous system.
Precisely. So actually, the way I started was when I was in medical school. I became very interested in neurodevelopmental disorders and autism in particular. After I met the first patient with autism, I was so struck by the clinical presentation, the complexity of the disorder, and immediately felt this immense frustration that there's absolutely nothing we can do. I was already used. I was going, an oncology ward or hematology, and where you would just like see a patient and immediately come up with solutions. They were all biologically based. There would be therapeutics, there would be diagnoses that were anchored in biology, and suddenly in autism there was like essentially nothing you could do other than prescribe some medications that have very broad effects and prescribe therapy of various sorts. And so it was like very frustrating. One, it felt like a challenge. At the same time, it was very frustrating. And as I was like doing my clinical training, I started spending my summers at the Max Planck in Frankfurt, in Wolf Zinger's department, who, as you know very well, has been a pioneer in gamma oscillation. So I started doing recordings from the cat visual cortex. And so suddenly there's like this paradox, this dichotomy, because I would spend time in medical school seeing patients, and measuring all kinds of metabolites in blood, trying to infer what is happening in the brain of those patients. And then on the other hand, I was at the Max Planck putting the electrodes inside the brain, recording directly as we were moving gratings on a panel, and like really making very strong, bringing very strong insights into biology. And I said, like, why can't we just do that for the human brain for these patients? There's no other way we're ever going to understand the psychiatric disorders unless we get access to these patient cells. And funny enough, is that precisely around that time, Shinya Yamanaka made the breakthrough discovery, or reported the breakthrough discovery, that you could reprogram cells, that you could take skin cells from an individual and push them essentially back in time through this genetic trick and turn them into pluripotent stem cells. And that was like a Cell paper pretty much around the time I was finishing my clinical training. And that was, essentially, seemed like a remarkable thing for me at that time. And I thought, how amazing would it be if we could just non-invasively make neurons from these patients, just neurons. I didn't think at that time about circuits or anything else. I just wanted to make some neurons from these patients and studying them directly in a dish to see what are the biological bases, the cellular basis of these conditions. And that's how it essentially started.
And then the value of that, then now you can take cells, you can take fibroblasts from someone with autism and controls, and you can try to see any differences. And we use this. My main area of work was on neurodegenerative disorders, but I started out as a developmental neurobiologist studying regulation of growth cones and synapse formation, and I showed that glutamate plays a role in the formation of circuits. So it's released from growing axonal growth cones onto dendrites that they encounter, and that glutamate kind of stabilizes the growth of it. But it's interesting because you're mentioning, so there's a big problem. How do you study human development? In rats or mice, it's pretty easy. You can get as many females pregnant as you want, and look at them in different. You can do genetic manipulations, but how do you get at studying human brain development, right? In a dynamic,
in a dynamic setting. Well, in a system where you can both like probe and manipulate those cells, because just getting the tissue, post-mortem obviously has not been incredibly, it's been useful to some extent. And I think some major insights in neurology came from studying post-mortem tissue, think Parkinson's disease and substantia nigra. But when it came to psychiatric disorders, there have not been major breakthroughs in post-mortem tissue. I mean, there is a genetic signature, beautiful genetic signature in most of these conditions. Clearly, there's pathology in that brain. But it's been very difficult to extract therapeutic opportunities out of that are, and so I think it's really important to be able to have systems where we can manipulate, where we can get causality to a large extent, and that has to be done with patient cells. And so it's been really exciting because in the beginning, as you can imagine, things were like very difficult. Nobody wanted to believe that this was possible. I mean, I remember, I was finishing my MD and wanted to get a postdoctoral fellowship to come to Stanford. And I started applying for postdoctoral fellowships. There were a lot of postdoctoral fellowships for autism research at that time. Autism was starting to become, for the first time, an area of like topic. The government was already started actually to fund a lot of autism research. And I remember applying for fellowships and thinking, oh, everybody's going to say, this is so obvious. Like, of course you should make neurons. And it was just like devastating. I mean, I have to open up, I guess, like those fellowships again, but it was like devastating. I mean, people were saying, this is really makes absolutely no sense. You're going to take those skin cells, turn them into stem cells, and then you expect to be able to turn them into neurons and even find something abnormal. I mean, the defects will be gone. In a way, all criticism is good. It made us think very carefully at that time that we have to choose the disease really, really well, that the first proof of concept has to be proof of concept, but a predictable one where you would expect something else. And actually has to do with calcium, because we were starting to talk about calcium. But essentially, the first neurons that we've made from patients were from patients with a rare form of autism and epilepsy called Timothy syndrome, which is very, 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 very rare. And we only probably know, there probably are 50 patients worldwide, but 20, 30 probably alive at this point. But the patients have a mutation, a single point mutation in the L-calcium channel CAV1.2. And it's a gain of function mutation. We knew that because, of course, you could do hex cells experiments, heterologous expression, and a little bit more calcium was getting into the cells. But these are voltage-gated calcium channels. So if you really want to study what they actually do, as you know, you do need to study them in cells that are electrically active. So cardiomyocytes and neurons.
Yeah.
And so we thought that this would be a great proof of concept. And so I came to Stanford in 2009 and joined Ricardo Dolmetsch's lab. It was my postdoctoral mentor. I had an incredibly visionary mentor at that time. It was like an exciting time, like thinking that we can make these neurons. And within maybe 4 or 5 months, we finally got the first neurons. And I still remember the night when I did the first calcium imaging that actually worked, which was with Fura2, by the way. It was a Fura2 experiment, super late at night. And I loaded the control cells and depolarized them with KCl, and I could see a nice spike, and it kind of like came back, and you could see it by eye. And then I loaded the patient cells. There were already neurons that we were deriving. And I just like couldn't believe it, because you would look at the movie and you would see that calcium like went up, but it was like not coming down. I mean, literally, you could see it by eye. And I was like, this is not possible. So I ran another cover slip and another cover slip and went through the night running cover slips and analyzing the data and showing it up like early in the morning to Ricardo. And indeed, you could see this beautiful phenotype where calcium would go up and come down in control cells, and in patients would go up and then it would stay for longer, which was just like incredible to see a phenotype like that. So we knew it was possible at that time. And of course, then came many, many months of like, doing all the experiments that are needed, all the characterization. And then subsequently, we published one of the first reports of human neurons derived from patient cells where you could find phenotypes. So that was like certainly very exciting.
And that was in 2D cultures?
That was in 2D culture. So at that time, most of the methods that we developed were at the bottom of a dish. And, for Thymitis syndrome at that time, it was more than enough, right? I mean, you just needed cells at the bottom of a dish. But there was one issue which like bothered me quite a lot as I was a postdoc at that time. And that goes back to your comment about human brain development. And, we were making cortical neurons. So every time we were staining the cortical neurons from markers, they seem to be deep layer cortical neurons. Most of them, the majority, the great majority, 80, 85%, 90% were deep layer neurons. Very few upper layer neurons. And of course, in cortical development, deep layer neurons are formed first, and then upper layer neurons are formed last. So I realized, well, then I should keep the cells cultures for longer, right? But of course, in mice, that process takes like 7, 10 days. But in humans, it turns out, upper layer neurons are made like at 24 weeks of gestation, 25. And you finish that at 27, 28, which is, way more than we could keep in culture. So I started to actually try to keep the monolayer cultures as long as possible. And as you know, when you do monolayer cultures, you keep them for like a few weeks, but at one point, they kind of like peel off, like the laminin beneath it kind of like runs off. And so I devised like all the strategies to replate the cells. So I would replate them on another cover slip, keep them another month or two. And even through the most heroic efforts, I could not go beyond the 100 days of keeping them in a dish. I mean, 100, 120, it was like very, very difficult. And so there were all these discussions at that time that corticogenesis is actually stalled in vitro. But I was convinced that it was stalled because we didn't allow it to progress, that the timing of development is probably conserved. And so you will need to wait for longer for those radioglia to start making upper layer neurons. And that's when we started playing for the first time with 3D cultures. Because at that point, I thought, well, why do we keep plating and replating? This seems to be the culprit here. You replate, enzymes, neurons are unhappy, laminin, maybe I'm not putting the right extracellular matrix, who knows? And so what we did is like one day I ordered low attachment plates. So forgot exactly, it was like a company in Germany that was making these plates that are coated, so the cells cannot stick. They have a polymer that they coat. And I got them, they're like very expensive. And essentially took the cells early on in development and just transferred them as balls of cells, like aggregated them, transferred them, almost like people were doing with neurospheres for many, many years. Right, as you know, kind of like use the same approach. And I was like, of course, to a large extent was inspired because Yoshiki Sasai, also in Japan at that time, was showing this beautiful experiment where you will be able to, for instance, recapitulate interesting aspects of like optic cup development in 3D. If you were to keep this, he was doing some cultures there, what we would call today 2.5D, because they were like kind of like raised, but they were still sitting down. But I moved into the low attachment plate. And I was like expecting, everybody was saying, like, I was like chatting with people in the cell culture room. Everybody was like, oh, they're going to disintegrate very soon. And a week passes, 2 weeks passes, 3 weeks, several months, and they're still there. And 6-7 months later, they were like still there. So then we took one, we sectioned it, and then we stained. And, remarkably, there were upper layer neurons in them.
And so one issue with that, Sergio, is that with neurospheres, generally, if they get so big, the cells in the middle start degenerating because they're not getting enough nutrients.
Yeah. So what we also discovered was like, with neurospheres, I think like the classic neurospheres, the proliferative capacity of those cells is immense. So they proliferate very, very fast, and you have to passaging them and cut them and everything else. Here, actually, they proliferate very, very slowly. And I think there's some sort of like homeostatic equilibrium because they grow to about 3 to 4 millimeters in size and then they stop.
Yeah.
And then of course the next question was, well, if they go there, and Ben Barris was in the department, was like the lab next door, and Ben, who unfortunately, passed away, very slowly became my other mentor at Stanford, and really was like a very close mentor and friend as I was starting my lab at Stanford. And
so then you started to have mixing glial cells in, or what?
Well, then Ben used to like come. I still remember him. I was like doing calcium imaging sometimes at night, and Ben would always leave very late. So he would like leave at like 2 a.m. and kind of like sing on the hallway and see the light on in the room where I was doing. And I would say, "What are you doing here? What type of experiments are you doing?" And so, well, we're trying to like get this human neurons. So I said, "I don't care about your human neurons. If you don't have astrocytes, it doesn't matter." And I said, like, look, we'll look for astrocytes. Just let me make sure that we get some neurons. And of course, that became a joke, but it also became an objective that we should look at astrocytes. And actually, together with Ben, we started characterizing astrocytes, and we realized that once corticogenesis, neurogenesis was complete, they would start actually making astrocytes.
Yeah.
And the astrocytes would be non-reactive. And then we characterized them. We had a beautiful paper together, just the last paper that I published with Ben on astrocyte maturation over a long period of time. And so it was quite an interesting time. And that's the time when I actually started my lab at Stanford. And the kind of like the field of three-dimensional cultures exploded. And so suddenly there are like many, many approaches of making 3D cultures and trying to leverage self-organization in these cultures. And initially, we primarily made Cortex. So we just focus on Cortex, cortical neurons, cortical astrocytes, various aspects. But of course, as you know, most of the interactions in the brain arise from, interactions between brain regions and between different cell types. So just think about the cortex. There are obviously a lot of excitatory cells, and perhaps in humans and in primates, there are even more interneurons that there would be in a mouse, but most of those interneurons actually come from ventral parts of the forebrain, have to migrate all the way to the dorsal forebrain. So when we were making the dorsal forebrain or the cortex, we were just making that. There are almost no interneurons in that preparation. Of course, that developmentally is not appropriate. How do we solve that?
You'll end up with seizures in a dish, or in a 3D culture.
So then we started thinking we need to somehow introduce those interneurons. And if we were to introduce them, we should introduce them in a physiological way. And that was like the first time that we started already to recognize that there are like remarkable self-organizing properties of these cultures. And so we came up with the idea at that time that we should actually fuse organoids that resemble different brain regions in what I later, actually, like a few years later, named an assembloid. And so in an assembloid, you put together, but it's more than just a co-culture. There is an emergent property from that interaction. And so in this case, we started planning, let's try to make a ventral forebrain and fuse it. And so while some people in the lab were working on the ventral forebrain, we started thinking, how are we going to fuse them? These are quite large. I mean, they're 3-4 millimeters in size. We kept thinking maybe we have to devise some special wells. At one point, we thought, oh, let's get some electrodes and so like impale them and get them in close proximity. Somebody else said, oh, let's get like some glue, like biological glue, and stick them together. But then how are they going to move from one side to the other? And then somebody in the lab came one day and said, look, it's actually very simple. If you make them, you put them at the bottom of an Eppendorf tube, and they fit exactly at the bottom of an Eppendorf tube, two of them, and you leave them there overnight, by next day, they're essentially fused because they love to stick to each other. And then you can pop them out of there and then just watch them. And so, and we started to do that and started imaging. And it was like really remarkable at that time. I remember we had lab meetings where we would just play movies of the cells moving, like for 2 hours. We would just like watch movies of interneurons moving. And Fikir Birey, Jimena Anderson, Chris Mackinson were like in the lab at the time. All three of them now have their own labs at Columbia, at Emory. But I remember like we were sitting in lab meeting and kept looking at these movies and seeing interneurons, which, as you know, move in a very peculiar way. They actually undergo nucleokinetic migratory steps. So they don't just crawl on the surface, but rather they almost like jump.
Yeah.
And so we started watching them. How are they jumping? Where are they going? What part do they prefer? What do they do once they arrive? So it's really, really exciting at that time.
Sergio, you want to just kind of give a little bit of technical comments on how you go about from the pluripotent stem cells generating, say, cortical neurons or different regions.
Yeah. So actually, that's most of what we spend time in the lab is figuring out the cocktail. Their secret recipe. Well, secret. We share it like right after, but kind of like figuring out, what is, the perfect cocktail to make a brain region. And usually, we're always inspired by developmental biology. So when we try to make a new brain region, we essentially like, look, think about it, like the ventral forebrain is known that one of the driving morphogen forces that makes interneurons is sonic hedgehog, right? I mean, all graduate students, undergrads know you try to make any structure in the part of the ventral part of the nervous system, you have to add sonic hedgehog. And actually, that's pretty much what we did. We took an organoid that was essentially, making a dorsal forebrain and just added sonic hedgehog. And suddenly the fate will change and they will become more ventral.
I had one kind of a permanent scientist in my lab, Pam Yao, and she started playing around with sonic hedgehog and our primary hippocampal neurons, and she had this interesting finding. She used these, they're called Campo Chambers. They have these little tracks. You have two chambers, and there's these fine tracks between them. You plate the cells in one chamber, the axons grow through the tracks to the other side. So you've got the cell bodies and dendrites physically isolated from the end of the axons.
Okay.
And she had this really interesting finding. Well, first she found that patched and smoothened were in the dendrites and not in the axon. So that was weird because she found that the sonic hedgehog was promoting axon outgrowth. And then she found if she adds sonic hedgehog only to the growing axon, it doesn't affect them. So we were thinking that this may be like an interesting way of like transneuronal during development, transneuronal communication, yeah.
No, totally. And I think that's a great example how to do this. And the other, is there background noise that you hear? Or? No? Okay. So the other, of course, like once we put them together and, we saw that interneurons were moving, the first immediate goal was we need to look at connectivity across brain regions, right? Because we want to see like most of the communication across brain regions is through axons and synapses or release of neurotransmitters or peptides. And so the first circuit that we actually built was cortical striatal. And so we started making a striatum. And that involved, of course, like using a bunch of new factors, looking at like how the human striatum is developing. Like we discovered that there's an interesting retinoic acid receptor that is expressed primarily in humans, finding an agonist for it, adding it in. And Yuki Miura in the lab was the one like really driving the work. And then he came up with this beautiful protocol of making, striatal organoids that have medium spine neurons, huge medium spine neurons. They're human cells. You play them down, they spread out and they have all these tiny spines. And then
those are GABAergic neurons.
Those are GABAergic neurons.
Yeah.
And so then we fuse them with the cortex. And then, of course, the beauty of that circuit is it is actually to a large extent unilateral. Like the cortex, part of the cortical neurons will project into the striatum, but the striatum doesn't project back. I mean, it comes back through a loop.
Right.
And so we put them together and then we started watching. And of course, what happens over the next few weeks is the cortex projects in the striatum does not project back. And then we thought, okay, well, we maybe we can do an even more complicated circuit, which we thought at that time would be almost improbable to work. We thought about like building the descending cortical spinal pathway, whereas cortical neurons connect with motor neurons in the spinal cord, and spinal cord neurons go out and control muscle contraction. And there are some interesting aspects of that circuit because in humans and in primates, in primates, there's one synapse less than in rodents. So cortical neurons cortical spinal neurons from the make synapse directly on motor neurons rather than through an interneuron as they do in rodents. Huh. So there was kind of like an interesting also level of evolutionary reason to do that. And so then Jimena Anderson in the lab spent several years perfecting a recipe for the spinal cord that would be enriched in motor neurons, so more like a ventral spinal cord. Yeah. That would have 10, 15% motor neurons, but a lot of the other cell types that you have in the spinal cord. And then we put them together with the cortex. And, I honestly thought that all the odds were against us. I mean, the neurons that are cortical spinal are a tiny fraction of like layer 5 neurons. Motor neurons are like 10% of all the cells in the other one. I thought that the probability that these two will find each other will be very, very low, if at all possible, when you have millions of cells in each. But then we did rabies tracing and a Cre recombination and found that the majority of the cells that are projecting are actually deep layer cortical neurons that express CTIP2. And then even more interestingly, then we made a bowl of human muscle. So we took a myoblast or iPS-derived muscles, aggregated them, and then fused them to the spinal cord. And then although motor neurons are 10% of the population of spinal cord, they're 90% of the cells that leave the spinal cord. So they're very eager. They recognize the muscle and they go there. And then you have these 3 parts that have essentially self-assembled because, we're not providing any instructions. We're not telling them where to go, how to do anything. There are some, I guess, like minimal rules of assembly that are present that come with the parts to a large extent. And once you do that, then the muscle will start contracting spontaneously. And then, of course, you can do optogenetics and now optogenetically stimulate the cortex and trigger muscle contraction 2 synapses down. And of course, you can do a lot of pharmacology there. And we've been using this model for studying a lot of disorders, especially actually for studying polio and non-polio virus-related infection, which are quite interesting. They're, as you know, like the receptor for polio virus is actually only present in primates. It's a little bit more difficult to study in rodents. There are all these non-polio viruses that cause really severe paralyzing encephalitis in children. We don't have a vaccine for it. There's very poor understanding. So it's been really very useful. This is collaborative work with Jan Karet's lab here at Stanford, trying to understand the biology, the pathophysiology of these conditions. So that was interesting, but the main
there's other viruses too that seem to affect the developing nervous system, a lot like Zika, right? That was
absolutely, yeah, and that was actually like beautiful work that was done by Guo Liming and Hongjun Song at UPenn, who really did like pioneering work in.
Actually, they were at Hopkins. They were at Hopkins where I'm a.
Or Hopkins, but work.
Yeah.
Yeah, you're right. Yeah, absolutely.
I had
I still remember
Wally,
seeing
I had her on one of my
data.
podcasts
Oh, okay, good.
last
Yeah,
year. Yeah.
yeah. And so I think so that's how that was happening, and of course we've built a lot of other circuits, and we're soon going to publish a few other circuits that are even more complex, where we can put 4 parts and not just like a linear arrangement, but actually like loop circuits as well, and all to a large extent leveraging the self-organization. And I think there's a huge opportunity in trying to understand what are the rules of assembly for some of the circuits, because and I always like say this, I guess like being trained as a physician, or like the physiology that usually like taught in medical school is that like the human body is a machine. So if you really, it's almost like an engineering perspective, where if you really want to understand like an organ or a part, you should be able to know every single part of that organ, every single component, and be able to put them together and kind of like know all the rules. So, to a large extent, I kind of like lived under, I guess, this shadow of a perspective that if you want to build a circuit outside of the human body from parts, you better know really well, the rules of assembly for each of that. What we did forget is that in biology, there is this remarkable force of self-organization that builds brains and livers and guts over and over and over again. Of course, our brains are very different from each other, but to a large extent, they're actually remarkably similar. And considering how complex they are, it is still remarkable that evolution has found these ways of building these complex organs over and over and over and over again from parts.
Yeah.
So those forces, that force of self-organization leverages instructions that are coming with each of the part. So when you make the interneuron and you make the pyramidal neurons, they each come with signals that will help the cells find each other. Yeah. And even if you don't know those signals, the cells will still find each other as long as you provide the minimal conditions for that to happen. And that's what we've been doing. We've been essentially hunting for the minimal conditions that we have to provide ex vivo for those processes to be recapitulated in an in vitro and then use them for disease. And as I was mentioning before, the whole motivation behind my work has been understanding disease. I know my lab is, I guess, to a large extent, like known for developing tools, but I actually never wanted to be a tool developer. That was like never, kind of like my goal. If anything, and people in my lab know, I hate building tools. I don't like building tools. I like applying them. I like to ask questions about biology, and I like asking questions about like medicine and finding therapies for disorders. And so most of the work in the lab that has been really in parallel with developing all these methods has been to apply them to understand disease. And we've built, dozens of models for various neuropsychiatric disorders, probably Timothy syndrome being one of the most advanced, because for this disease now we've understood so well the biology of these conditions. And I've only described the early experiments, but we've gone so much farther that a therapeutic opportunity started to arise from those experiments that we were able actually to test systematically. And, I still very much hope that we'll be able to contemplate the clinical trial in the next period. And I'm so lucky to be here at Stanford and have, residents and physician scientists who are interested also in the translational component. I don't see patients anymore. But that really is like what is emerging as the, what I think personally for me, the most important contribution, which will be to attempt translation for neuropsychiatric disease.
Yeah, and there's tremendous value in a lot of different aspects of these organoid assembloid systems. One is, in theory, you can reduce the number of animals you need, right? And then two, you're working with human structures that a lot of drug trials fail, that are tested in rodents and then they don't work in humans. And my understanding is the FDA is even considering using these 3D culture organoid things as kind of part of their approval process for drugs.
Yeah, no, you're absolutely right. And we've seen this quite remarkable like change of mindset, I would say, in what it takes to develop therapeutics for, especially for neuropsychiatric disorders, where I think we've had the most challenges in terms of translation. And so we've all seen that the FDA move towards like the FDA 2.0 version, where essentially animal models are not necessarily required for approval of a drug. Now, again, it doesn't mean that we should not use animal models, and we've been using in combination animal models and human cellular models, but there are instances where the biology of a disease is not recapitulated in an animal model, or some of the phenotypes, and so it's challenging. We're stuck, and the question has been, well, now if some of that biology is recapitulated or can be identified in human cellular models, will that be sufficient? And I think that remains to be opened. And in fact, the NIH director has had a special advisory committee, which I had the honor of serving for the past year, discussing precisely what are going to be the alternative models. And that report hopefully will come up like very soon. What are going to be the approaches that we should be using? And I think ultimately, we will see where experiments take us, all in terms of translation. But I think it's really exciting to have all these models available: human models, animal models, primate models. I think it's kind of like it's the best of times for biology, really, to a large extent.
Can you talk about your recent paper where you're looking at the integration of human cortical organoids into the rat cerebral cortex, which is kind of interesting?
Sure. So, as I was mentioning, most of the work that we've done was certainly in vitro. And we've been able to build ever more complex circuits in vitro, there's no doubt. But we were always aware that there are limitations of doing experiments in vitro. As you can imagine, the cells don't have a blood supply. They're not receiving a lot of nutrients. Maybe even more important for us as neuroscientists is they're not receiving activity. There's a lot of activity happening in the nervous system that, as we know, is shaping the maturation and the activity. I mean, pioneering work from Carla Schatz showing how important activity is during development. And, there's no way to model that in vitro. And we do know that it does matter, right? Yeah. And the other thing is, like, we also notice that just generally, no matter how long we keep some of the cells, even if they recapitulate a lot of the timing of development and a lot of the molecular features, the cells never reach kind of like the electrophysiological maturity that you would expect from a neuron. So it's very difficult to get a neuron in vitro that will have a resting memory potential of -70. I mean, it will be more like -50, maybe even -40. They will not fire as much as you would expect them. So we were aware for a while that we're missing a lot of aspects. So we thought, could we actually put them in an in vivo environment and perhaps leverage some of some of the, I guess, like cues in the host for maturing the tissue. And as you know, people have been doing transplantation of human cells and human neurons for like decades now, right? Like since the late 70s and 80s, especially as immunocompromised animals became available, that allowed to do even more transplantation, because now the human cells will not be rejected for the graft. And there's been beautiful work done in transplanting individual human neurons in circuits and embedding them. But what we were envisioning was to essentially build animals where you would have a unit of human tissue, of human cortex, in a predictable position in the animal that would allow us to run assays and obtain enough integration in the circuit that we could actually run perhaps even behavioral assays. And so what we did is we knew it would be very difficult. We've done experiments for years. I mean, this has been going on for like 8 or 9 years in my lab. Initially, we were doing transplantation into the adult cortex. So we would get immunocompromised rats, take an organoid and place it into the rat. And it was always difficult for the graft to survive, or if it was surviving, it was not too much connected to the rest of the host. And that's not surprising, because at that time, critical periods for the rats are closed.
Yeah.
There's a lot of myelination. Most of the pathways are already, formed. You don't get too much in terms of like new connectivity. So then we thought, how about going early in development? And so we started doing transplantation before P7, where the critical period for the cortical thalamic cortex is closing. And we transplanted them into the somatosensory cortex of the rat, so the barocortex. And the way we did this, and this was work done by Omer Revach in the lab, who's now actually has his own lab in Israel, and Kevin Kelly and Felicity Gore. What we've actually done is like we've transplanted the intact organoids into the somatosensory cortex of the rat without removing rat tissue, and then waited. And we discovered almost by chance that you can see the graph by MRI. And actually the border of the graph. That made a huge difference for us because suddenly we were not kind of like at the mercy of like time and pathology, where you would wait for like 5-6 months, get the tissue from the rat, and then see where is the graft, how did it grow. But now we would non-invasively go, and even a few weeks after we transplanted the graft, we would go to the MRI and see where's the graft. Is it growing? Should we do the angle of injection differently? So we perfected the surgery very quickly, so we could get very good integration. And then after 4 or 5 months, you would essentially have a unit of human cortex that would represent about 1/3 of a rat hemisphere. And it would go from pia all the way to the ventricle. And it was vascularized by the rat. We found terminals of the thalamus in it. So then we thought if there are terminals of the thalamus in it, then maybe there's thalamic activity. So then we could actually like open the skull to calcium imaging of the human graft and move the whiskers of the rat on the opposite side because the pathway is crossed, and obtain responses of human cells. So that shows us they were very well integrated. And why does that matter? Well, it does matter because now if you take out the graft, the human neurons, after 200 days, and you compare them to cells that were maintained in vitro from the same line, same differentiation, the cells have very different properties. Cells are about 6 to 8-fold larger in size. Resting member potential is -70 now. They have synaptic input. They have spines, because they have been in that in vivo environment. And we still don't know exactly what is it activity, is it growth factor, is it a combination, and we're going to figure it out. But it is quite remarkable.
So do you Tony was Corey, right?
Yeah.
And what about bloodborne factors that might be playing a role?
Absolutely. It could most certainly be that there are blood factors, and just in general, like the rat brain, the astrocytes in the rat brain could be secreting all kinds of factors. We just don't know yet.
Yeah.
But the reason why this is also important, because now you can actually even discover phenotype that you would never see in vitro under resting condition. So in Timothy syndrome, for instance, if you make neurons from patients and controls, and as you know, L-type calcium channels are very important for like dendrite development, for kind of like morphology. But you make neurons in a dish, and you look after like 200 days or something like this, you wait as long as you can possibly wait. You compare the neurons, they're indistinguishable, patients and controls. But if you actually depolarize them, so if you actually start depolarizing the cells in a dish quite strongly, then the patient cells will actually retract their dendrites and the control will like actually expand. But you have to do the depolarization. Now, here's the beauty. You can now transplant patient cells into rats. Don't do any stimulation, anything else. You come 150, 200 days later, you compare the morphology, and the patient cells are actually retracted in vitro versus in vivo. It's because the activity now is present in vivo. So that gives us another level of confidence. It shows us that with this transplantation methods, we can discover phenotypes that we would not be able to discover before. And so,
and you're also doing so Timothy syndrome's pretty rare, right?
It's very rare.
Yeah. And what about autism? Have you been?
Yeah. So for autism, the way we kind of like see autism in general is as a collection to a large extent of genetic disorders, many of them, and I think autism is one of them. And of course, most of them have multiple presentations. Patients with Timothy also have epilepsy. They have schizophrenia when they're. So I think it's been very difficult. But our approach has been to go after disorders that are very severe, have an early onset, and the genetic mutation is very clear, and provides us with something tangible to go after. Of course, the problem is that the majority of the cases of autism are idiopathic. We don't know the cause. So those are going to be much more difficult to tackle moving forward. Yeah.
And could you briefly talk about your very recent Nature paper on using a CRISPR screen in assembloids, and you found this interesting thing with endoplasmic reticulum and calcium, which is interesting to me because we actually did a lot of work on that in relation to presenilin 1 mutations in Alzheimer's disease.
Yeah. No, and this is beautiful work done in the lab by Shanling Meng, who was very interested in understanding, what are the genes that actually associate with disease that impact interneural migration. And 20 years ago, there was this hypothesis that excitation to inhibition in the cortex is a driving force for autism spectrum disorders, primarily anchored in the fact that patients with autism have more epilepsy and then EEG findings and so on and so forth. And that has been, of course, around for a very long time. But the question was, and there's no doubt, there are forms of autism that probably impair either the formation or the migration or the integration of these interneurons into the cortex. But we now have hundreds of genes associated with autism. Which ones of those interfere with this? Is there a way of actually identifying? So that was actually the rationale for our screen. So what we did is we compile a list of neurodevelopmental disorders, more than 600. And then we looked at all the genes that are expressed in interneurons at any time during development and ended up with about 425. And then we did a screen for all those 425 with the genetically engineered IPS lines, trying to identify which genes interfere with the generation of cells and which genes interfere with the migration of cells. And it's a long story, and I'm sure people can read the paper if they really want for the technical details, because it was like really, a technical feat on like Shanling's side, who made at one point 1,000 assembloids and cut them one by one individually and took the cells that migrated and sorted them out to identify all the guide RNAs that have actually impaired. And she identified that about 10% of these 400 genes actually interfere with one of the stages of development. And some of them were very much expected. We knew that they were interfering with interneural migration, but one of them caught our attention because it was actually a structural gene of the endoplasmic reticulum. So the endoplasmic reticulum has not been classically associated with interneural migration. It's been suggested actually many years ago by Mary Beth Patton that it is actually, undergoing, movements during migration. And so we actually started watching, and Shanling actually started watching the ER during interneural migration. She discovered something quite remarkable, that almost the entire ER moves in front of the nucleus as they're making that jump. And now if you knock out Luna Park, that fails to happen in most of the jumps. So we think that impairs the migration of the interneurons. And I think this illustrates how, and that's why I think it's like so exciting, because we've reached a point where this truly inaccessible aspect of human brain development, because, look, interneural migration happens in the second trimester, third trimester of life, up to the second year of life in humans. Truly inaccessible stage of human brain development. How this truly inaccessible stage of human brain development, not only can we recapitulate it in a dish to a large extent, but we can even start mapping hundreds of genes associated with disease to understand which of those interfere with stages and even learn something new about the biology of the cells, which I think is probably some of the most exciting aspect of it. So, so that's kind of like, briefly the story.
So early on, Sergio, you mentioned that it's been fairly recent that there's a lot of money put into autism research. Well, the last few decades anyway.
Yeah.
But the increase in incidence of autism, actually, I was interested in this and did a Trends in Neuroscience article on it, that, okay, it tracks with maternal obesity.
Yeah.
And there is evidence that women, if they have obesity, type 2 diabetes during pregnancy, there's increased chance that their child will develop autism. It's still low, but it's increased relative to.
Yeah.
And so this suggests something to do with, to me, energy metabolism and the mTOR pathway. Like there's this like overnourishment almost.
I think there are many causes for autism.
Yeah.
I do believe that autism is not one disease, but rather a collection of conditions. I think probably some of the forms of autism will be related to this migration of interneurons and epilepsy, perhaps in these cases. Others will be caused by mitochondrial defects. Some of them will be caused by microglia dysfunction, right? Yeah. So I think what we will see over the next decade or maybe couple of decades, we will see a kind of like a biological dissection of these conditions, or hopefully we'll have clusters that are biologically inspired, which at one point will maybe even guide therapeutic development. So I think that is kind of like exciting.
In your organoids and assembloids, what's the energy source? Is it mainly glucose?
It's mostly glucose, yes.
I think it would be interesting to play around with ketones.
Yeah, absolutely, yeah.
And then finally, I know you're busy. Ethical concerns.
Yeah.
You want to make any
Oh,
comments?
absolutely. We've been, as you can imagine, some of these experiments, especially as we do transplantation experiments, are delicate for multiple reasons. They raise a number of ethical concerns. We've been actively discussing those as we were doing, especially before we were even doing many of these experiments at Stanford. We're going to have we're actually going to have a meeting on the ethical aspects of this, which is going to be taking place at a Silomar sometimes next year. We're going to discuss the ethical concerns, and I think it's very important that we engage philosophers, ethicists, lawyers, neuroscientists across the spectrum of neuroscience expertise to start thinking about these models very seriously. And then interface with the public as well. I think the public deserves to know what type of experiments we're doing. Yeah. I think the public deserves to know that the motivation behind most of this work is to tackle these devastating disorders. Psychiatric disorders still represent, the largest source of the largest cause of disability worldwide. At least 1 in 5 individuals is suffering from psychiatric disease. We don't have therapeutics that are curative for none of these conditions, actually. Yeah. So we will need to make progress. And we're going to need to do that being anchored in human biology. And of course, as the models are becoming ever more human, the more uncomfortable we feel. And therefore, I think these conversations need to take place. And, we already started, and there are like several papers that we've published on this, but I think these discussions have to continue as the models are becoming more advanced, so that we know how far one can actually go.
And of course, these same kind of approaches are going on outside the brain and different,
absolutely,
cardiovascular system, muscular.
Absolutely. And it's been quite interesting. Like one of the most interesting things is like I've seen in the last few years, now there are assembloids across the entire nervous system. I mean, there are like bladder assembloids, gut assembloids. It's quite interesting to see how people immediately found ways of like assembling, because I mean, that's a general principle in biology, that you generate different cell types in different niches because you need different signals, and then they come together. Otherwise, you wouldn't be able to make them at the same time. So immediately, assembloids became, the tool that enabled those interactions. You make things separately, you put them together, you look at an emergent property, a new physiological effect, a new disease phenotype. So I think it's going to be really exciting to be able to study this complex cell-cell interactions in the context of human biology.
Has someone done brainstem gut to try to make like essentially the vagus?
There are some efforts in that regard, but nothing has been published. Yeah.
Huh. Yeah, because in Parkinson's, there's this evidence that there's this retrograde.
Yes, absolutely.
Okay, Sergio, I appreciate you taking the time for this. I was once maybe as busy as you, but not anymore. I'm retired, so it's
well, it's great to have this conversation, and hopefully those listening will appreciate it too.
That's great. Sounds like you got a great lab with a lot of really energetic.
Oh, amazing! I've had a blast. The students have been just absolutely amazing. Yeah.
Good. Okay. Bye. Have good holidays.
Thank you so much. Thank you. We'll be in touch. Look forward to seeing the recording.
Okay. Bye.